Welcome to McGill University's Department of Pharmacology. We're here in the Hébert Lab, located in McIntyre Building, where cutting-edge research is being conducted to better understand diseases in the heart and brain to develop new, personalized treatments. Today we'll take you on a lab tour and introduce you to some of our graduate students who are part of our research team.
Hi, I'm Terry Hébert. I'm a professor in the Department of Pharmacology and Therapeutics here at McGill University, and I've been asked to tell you a bit about what my lab works on. We study a few different diseases in the lab. Some of them are cardiovascular, so heart failure, dilated cardiomyopathy, and some of them are neurological. So we're interested in depression, we're interested in anxiety, and we're trying to develop some better models to study these diseases, but at the same time use a lot of really state-of-the-art drug discovery tools in those better models. So we're interested in using things like patient-derived stem cells, where we actually collect blood from patients and turn that blood into a stem cell. And if we're interested in muscle disease then, so cardiac disease, we'll make it into muscle cells. If we're interested in the neuronal side of things, we can make those into neurons, right? So the idea eventually will be to kind of start to personalize therapies. Can we start with an individual patient and understand how the disease affects that patient, understand how to treat that particular patient?
So, how is this all done in the Hébert Lab? Well, it starts with the cell culture room, where we maintain our patient-derived stem cells, which are then differentiated into cardiomyocytes. Let's speak to Deanna, who's working on that right now.
Hi, I'm Deanna, a PhD student in the Hébert Lab, and I'll be going through our differentiation process where our stem cells are reprogrammed into heart cells or cardiomyocytes. The work that we do in cell culture is very important since the quality of our cells will ultimately impact the success of our disease models and our experiments. Our workflow, which takes about 25 days, involves adding factors that modulate the Wnt beta-catenin pathway, and this gets the cells to become cardiac progenitors. Then, once we have these progenitors, we'll use a unique property of cardiomyocytes to survive in conditions without glucose to remove any other unwanted cells, leaving a plate of cardiomyocytes.
So, what do we do with these cells once they're differentiated? Well, to talk about how we image these cells, let's go meet Jada in our imaging room.
Hi, I'm Jada, a master's student in the Hébert Lab. Behind me is the Phoenix, a powerful microscope that we use to look at cell structure and activity. Here's an example of cardiomyocytes, marked with a biosensor for calcium, which localizes to the contractile units of cells and allows us to gather lots of data one cell at a time. We're also able to image organoids, which are essentially three-dimensional stem cell-derived constructs, allowing us to look at cell signaling and gather information on different cell types, interactions between these different cell types as well. With all this, we can look at cell signaling in individual patients with the goal of distinguishing differences between these patients to allow us to move towards more individualized disease models as well as drug discovery tools.
Now let's talk to Dieggo, who's working on assessing the contractility of cardiomyocytes using the cardio-excite system.
Hi, I'm Dieggo, one of the PhD students in the Hébert Lab, and I'm here with the cardio-excite system, where we can look at two different functional parameters of our stem cell-derived cardiomyocytes, that being extracellular field potential, or EFP, and impedance. The EFP allows us to generate a curve similar to an electrocardiogram, where we see the initial depolarization of our heart cells through the QRS complex, and subsequent repolarization through the T wave. Impedance, on the other hand, acts as a really good measure of contractility, because a contracting heart cell would resist current differently pre- and post-contraction. Now, with both these measurements, we can directly assess the function of our cardiomyocytes to hopefully identify differences between healthy subjects and patients with heart disease.
But cardiomyocytes aren't the only cells being differentiated in the Hébert Lab. Let's talk to Dulce, one of the graduate students working on differentiating neurons from stem cells.
Hello, my name is Dulce. I'm a master's student here at Terry's lab. I'm working with iPSCs and differentiating them into cortical neurons. This is a process that takes approximately 2 months, and it involves several steps. The first step is to form embryo bodies, which are then transformed to neural progenitors through a process called neural induction. These progenitors have the ability to turn into many cell types from the nervous system, including oligodendrocytes, astrocytes, and neurons.
So altogether, you got to see some of the techniques we do here in the Hébert Lab, including differentiating heart cells and neurons to assess the functional properties of these cells. We hope you enjoyed it and learned a few things along the way. Thanks for listening.